Okay, let's pick up where we left off, shall we? Okay, so last time we left off, we were getting ready to talk about the monohybrid cross. And one of the things I wanted to do was I wanted to go through what Mendel went through. So I wanted to see what he saw and basically kind of reason through his data, through his observations, and sort of see how he basically kind of built it all. Now, one of the things with Mendel that I like with Mendel, and it's not necessarily just my bias. I mean, it is my bias, but it's not all my bias, right? Because it's true as well, is that Mendel is actually probably one of the neatest, cleanest illustrations of the use of the scientific method in scientific history. Now, it doesn't necessarily mean he created it. He didn't. There was pieces and bits and pieces that were being used throughout. A lot of the chemists had a lot to do with that as well. But when you're talking about like the 6-point scientific method, Mendel's experiments were like simple, clear cut, and basically he hit every single level of the scientific method. So it's like really a great illustration of how to use scientific method in order to do really good research, and how to do really good research. It gives you a lot of information, a lot of data, which is exactly what he saw, right? And so I like to sort of think of this as sort of like, since this is kind of like 1865 or so is when he published his paper. I like to think of this, and it's maybe a little overspeak, but this is my bias. But I like to think of Mendel as sort of being the one who kind of taught us how to do science. In a lot of ways, the way he did it, it was an illustration, an example for us to follow that kind of set up the 20th century. And as a matter of fact, when you take a look at 20th century research, you think of the rigor and kind of the way we sort of go about it. A lot of those were greatly influenced and helped by the way that Mendel approached his work in a very rigorous, sort of a very scientific and a very intellectually honest way. And so he kind of in a lot of ways sort of taught us how to do science well. Not that we didn't already know how to do science. I mean, we'd already been doing it, but a lot of it was observational. And a lot of it wasn't experimental. And that's kind of a different creature. You're just looking at stuff, collecting stuff. It's different. So Mendel kind of taught us how to actually sit down, do an experiment, and sort of go down all your points. So what I want to do is I want to basically on this side, I want to list our scientific method, and we'll take a look at and see how he kind of hit every single piece, right? So we learned about this in chapter 1, right? So our first piece of it, our scientific method, is our observation. And then from our observation, we generate a question. And then of course, from our question, we come up with our best guess, right? Our educated answer, right? That's our hypothesis. And then from our hypothesis, we have to generate some testable predictions. Those are those if-then statements. If my hypothesis is true, then I expect this to occur, right? Those sorts of things. And then once you get testable predictions, then you do some sort of an experiment to test those predictions. And then last but not least, from those experiments, you generate some sort of conclusion. Now, let's take a look and see what Mendel did. So taking a page out of TA Knight's script using pea plants, he generated, first of all, his parental generation, and he generated true breeders, right? So basically you have your true breeders. So he took basically, this is a purple. And he crossed this to a true breeder for white. So then, first thing he did was he collected data on his F1, right? Just like we saw with TA Knight forming that first village generation. And what did he see in his F1s? Yep, and they were all, in this case, purple. So everything's purple, right? So what's his first observation? F1 is all purple. So this is observation. What's his question? Why? It's always a good one. Yeah, what about white, right? So that the basic question is very simple. Where's white? Right, so that is his question. His observation is that all of his F1s are purple. Now you have two different answers to that question, right? Your two competing hypotheses. The first one is the blunders, right? The blunders say what? White is gone, right? Makes sense. Your blue and your yellow, mix it together, you get green, your yellow is gone, never to be seen again, right? That's what the blender said. Of course, the particulate guy said, no, white's still there somewhere, right? But maybe white is hiding, right? So there's your hypothesis, your best guess based on what you know currently. From each of these, you're going to be able to generate a hypothesis. So the first hypothesis for the blenders is if it's gone, then it's non-recoverable. Just like your yellow is non-recoverable if you mix it with blue paint, right? However, the particulate guys say, well, no, wait. But if it's hiding, then it's going to be recoverable, right? Notice the if-then statement. If the blenders are true, then you expect never to see it again. If the particulate people are true, then you expect it to see it come back, right? So notice you have testable if-then statements. Those are your predictions. So then what's next? So here's your predictions. So the next is going to be your experiment, which is what? Cross your two purples together, right? So you probably self them together. Let's go ahead and see what happens. Now what's going to happen is you're going to get an F2 generation. It's going to look something like this, yes? 3 purples. To 1 white. So that's your experimental data, right? Now what's your conclusion? Did you recover white? So then that suggests that your conclusion, your data, supports the particulate hypothesis. Very simple, yes? But wait, no, the nice thing about this is if you take a look at it, notice you've basically just in a very simple stepwise process, you've pretty much just gone through every single step of the scientific method in crystal clarity, right? That's the reason why I say Mendel is probably the best illustration of how to use scientific method and just kind of go through the process, right? Now, in the 20th century, we still use scientific method, but there's bits and pieces of it we kind of omit. And some of them we kind of short circuit a little bit, and we'll use some pieces and not others and things like that. But for the most part, this is the best example of just the sort of full-fledged process of the scientific method. Now, let's talk about this for a second. Mendel's now just looking at purple and white. But other than just basically your discrete conclusion, which is a big deal. I mean, after all, you just basically discredited the blending theory, right? Your data is not consistent with the blenders. So the blenders got some explaining to do, right? But there's more to this, right? Because there's a lot of questions here. Now let's just kind of reason our way through this. First thing we want to do is insert another slide. Because what I want to do is I want to basically build a glossary of terms we now know because we have the book, but that we didn't have back then. These are things that Mendel had to figure out from his data. Mendel's ideas. Glossary of terms. First of all, before even beginning, remember Mendel started off with this idea because he was a particulate guy. So he started off with this idea that there's a packet of instructions, a particle, if you will, of inheritance that's passed on from one generation to the next. This particle we call a gene. So now we know a lot about that particle inheritance, right? We know a lot about genes thanks to the 20th century. Mendel, however, did not have any of that 20th century. Wait a minute, that's not the button I wanted. That's the one I wanted. Good, good. So just jumping on. So Mendel didn't have any of that, but this particle of inheritance is what we now refer to as a gene. However, Mendel's a smart guy. So he's taking a look at this. You got purple flowers and white flowers. So in one sense, your gene, your particle of inheritance, is governing a particular trait. That trait is what? It's flower color. But what he noticed was that there's two versions of this. You have a purple version and you have a white version. Is purple flower color? Is white flower color? So you have two different versions of the flower color. So for this one particle, you have two different versions of it. Those versions we later called alleles. For instance, you have the purple versus white allele. That's before he even started. You don't have to do a single cross to figure that one out. I mean, you've got like a little garden full of purple flowers and white flowers, right? However, let's start digging into this. Let's actually dig into his F1 data. So what he sees is all purple. Now he's a particular guy, right? And his conclusion basically said that white is there. But do you see it? But do you have white? But do you see it? So you have two different versions of this gene, purple and white. Obviously F1 has purple, right? Because you can see it. But now we're basically saying it has white in there too. So you have purple and white together, but you only see purple. So these two versions must have different strengths to them. So when you look at this, then he basically reasoned out the concept of the dominant allele. A version of the gene that is expressed in one copy You also notice that the white one is a recessive allele. This is a version of the gene that is expressed in 2 copies and masked by the dominant. Oh, but wait, let's keep looking at this for a second. I'm noticing something. I'm going to take a look at this flower right here and this one right here. If I were to take those 2 flowers, put them next to each other, do the old shell game thing, and ask you, can you tell me which one is which? Can you tell the difference between a parental and an F1? That is this parental and this F1. Can you tell me the difference? If I were to sort of switch them around on you. Point to the one that's parental. Why? Because what do they look like? They're both purple, right? Okay. But think about the way they behave. Do they behave the same way genetically? No. Why? Yeah, right? Because, or if you don't know anything about alleles yet, right? The idea is this one is a true breeder. It breeds true for purple. This one does not. So that means genetically they behave differently, but they look the same. So what do we call that? We find the difference between phenotype and genotype. Phenotype is what you look like. Based on your alleles. Genotype is your allele makeup at a gene. Generally speaking, in a simple world, your genotype has a one-to-one relationship to your phenotype. But in reality, phenotypes are much, much more complicated than that. And that's kind of the reason why we have an entire class on genetics. We learned about this in the 20th century, is that your phenotype, as complicated as it is a combination of genetic and non-genetic factors. And so in genetics, we kind of plumb the depths of all those different variables. I always like to tell students, think of yourself as like a diamond. Each human phenotype is like a diamond with like an infinite number of facets. And each one of those facets is a particular variable that adds to your phenotype. Some of those facets are genetic. Some of them are not genetic, but they all add to your phenotype and creates a lot of complexity in this, right? The way this looks. So not only that, but then if you kind of get to the idea of genotype and kind of to follow up on what Shane mentioned, is you can reason this out, right? Think about it. What does it mean to be a true breeder? Yeah, all you have is purpleness, right? So all you have is information for purpleness. Now, if you know you've got basically 2 copies of this, then that means that whatever you have is 2 copies of purpleness, right? So typically in genetics, we denote that it's a dominant as uppercase, right? So we know it's dominant because we just determined that the dominant allele, in this case purple, is hiding the white, which makes it dominant. White is recessive. The dominant is always uppercase, recessive is always lowercase. So if you're a true breeder for the dominant type, then that basically means you're going to have 2 alleles, as Shane said, of the actual purple allele. Because that's all you've got to give, right? That makes you a true breeder. White, on the other hand, is going to have 2 copies of the recessive allele. And that's going to make it a true breeder because that's all it has. So then what happens is this individual parent will give the big P, this individual parent will give the little p, and what you have here in the F1 is basically an organism that has 1 of each, which makes sense, right? because you've got the dominant purple allele, which is covering the recessive white allele. The white is still there, so it can come back around if you put in the right combination. So what Mendel basically did was figure out the different genotypes. So the first one is homozygous dominant. This is where you have 2 copies of the dominant allele. So for instance, that. You also have homozygous recessive. Where you have 2 copies of the recessive allele. And then last but not least, we've got 1 of each. Your heterozygous. 1 dominant allele and 1 recessive allele. This would be your genotype. Homozygous dominant, homozygous recessive, or heterozygous. Phenotypically, however, it's going to be a little bit different, right? So if you take a look at the phenotype, then basically this guy would have the dominant phenotype for whatever A is, right? This guy here would have the recessive phenotype for whatever little a is. And big A, little a, because it is dominant and a recessive, is going to have the dominant of whatever that phenotype is. So you can see already that you can set up and establish some relationships between phenotype and genotype. But notice you get all that by simply just taking a look at this and just thinking about it a little bit and asking some questions. That's actually quite a bit of information from such a simple little cross. So very simple cross, very simple setup, and you're able to actually set up a lot. Now, it's important to understand when we have our like little glossary here, that all these terms here on the left, Mendel didn't have. If you actually read Mendel's paper from 1865, which you can, because it's still available. I actually posted on the genetics website for students who want to read it. It's actually kind of fun. He never mentions any of these terms, because it's always important to remember, because in 1865, that predates most of our knowledge. Like for instance, all the information from chapter 10, Mendel didn't have. Chapter 11, Mendel didn't have, right? So all that stuff about mitosis and meiosis, that makes you smarter than Mendel, because he didn't have any of that. So he didn't know about chromosomes. That hadn't come together yet. So what he was following was just information. He was just saying the information that's in charge of this trait. That's why I like to use that as terms of a packet of instructions, not as an analogy, but because that's the way he thought about it. He was following information flow from one generation to the next. And this is basically what he saw. So all these terms came later. Afterwards, in the turn of the century, when he got rediscovered, then those guys basically came up with all these names and terms. What you'll see in Mendel's paper is actually descriptions of these. So you'll hear him talking about something and describing something, and you're like, he's describing a recessive allele, right? Or he's describing a dominant allele, or he's describing homozygous dominance, right? That's what he's describing. And it's actually kind of fascinating because you can see the elements, the kernels of most major branches of genetics actually laid out in Mendel's paper. Like he even talks about the quadratic formula of what later becomes the Hardy-Weinberg equilibrium, which is part of population genetics. And that came decades later, almost half a century later after Mendel. But you can hear him talk, there's a little bit of it. You can see a little quadratic formula in there. And it's like he was even dancing with population genetics at that point. So, I mean, just about every little kernel of genetics, you can actually read just a little bit of it in Mendel's paper. And then later on, what happened is succeeding generations took those little kernels and then blew them up and then developed them into population genetics and stuff like that. This is one of the reasons why the guy has a chapter of his own. Yeah.
How was he able to know that, what did you call it, like the pure purple, the P? For the original purple, how did he know since like when you breed it with white, you can't see the white, how does he know he's even starting out with the proper purple? Because he had to have just been breeding them the planet.
Yeah, because so what he'd done is he had like TA Knight, he developed the true breeding strains.
Okay.
And then he'd been breeding these strains like in perpetuity for a long time. So he'd had, before he started, he had multiple generations of true breeders. So he could go back and say like, this purple plant is a descendant of 12 generations of purpleness.
Yeah.
And there's only purpleness there.
Yeah.
And so what that does is just like with TA Knight is that eliminated all those variables. So it really simplified the system and said, okay, whatever is going on here, I don't know. But the information for purpleness, this is all it's got because this is all this family lineage passes on. Make sense?
Yeah.
Now that part of it is inductive, right? Because the idea of inductive is if you make that many observations over a period of time, then the idea is that it's consistent. You can say like, okay, these guys are true breeder now for purpleness. Why? Not because they just spit out purple, but because you've had multiple observations over time that everybody in this lineage or in this family only produces purple. So there's no reason to suspect that the next generation isn't going to produce purple. Does it make sense?
So like you started and then you bred purple, you just get rid of the white ones and then purples together.
Mhm.
That process.
And then the purples, and then once it happened, you basically the purples would get purple, and the purple would get purple. And if he had a pollutant in there, eventually you'd see a purple, and a purple would produce white, and he would trash that, and you just stick with the purples and the purple. Similar to the way Jacob did with the sheep, which is the reason why I lead with that story, is because he was doing the exact same thing with the sheep that Mendel has to do genetically with the plants in order to create those nice strains. So basically he kind of got a lot of information just from that simple cross. And of course we know that once he basically bred this out, he generated his 3-to-1 phenotypic ratio, but there was more than that. He also found a 1 to 2 to 1, what's called a genotypic ratio, right? So basically, whenever we look at this, we have what's called a phenotypic ratio. Remember, this is the way you look. This is purple to white, or dominant to recessive. And then you have your genotypic ratio, which is your homozygous dominant to your heterozygous to your homozygous recessive, which he found to be 1 to 2 to 1. Let's take a look at this here for a second. So this is what he did, right? So here's his parental generation. Took a true breeder for purple, crossed it to a true breeder for white. Then he got his F1 generation, which was all purple. And then he got his 3 to 1. So here's your 3 to 1 phenotypic ratio. But he didn't stop there. He went to an F3. Right? Because he said the same thing. He said, well, wait. If I can't tell the difference between these two purple plants, then how do I know that all these purples are the same? If I can't tell the difference between a true breeder and a non-true breeder, then how do I know which one's which? Well, basically what he did was he selfed each class of plants together. In one, he found that you had a true breeder. For the other two purples, he noticed that you didn't. You had a 3 to 1 and you had a 3 to 1. So these guys were non-true breeders. And then for the white one, these guys were true breeders. So when he did this, he got a 1 purple true breeder, 2 non-true breeders to 1 white true breeder. So these are the non-true breeders, and these are the true breeders, based on what he got in that following generation. So this is his genotypic ratio.
That means that a white recessive will never be able to give purple.
Correct. And a purple true breeder will never be able to give a white.
define that it becomes a tribute, so I guess
Yeah, because this is because these guys will produce, these are non-true breeders. The only way you can get white is out of those guys, right? Which is basically kind of what, I mean, if you think of this like in the old Jacob sheep story, what he was given was the so-called not so great sheep, right? The brown spotted sheep, which would be these guys here. But 2/3 of those would produce, at a small percentage, about a quarter of them, would produce offspring that are white. Some of them not. Right, but some of them, yes. And you could imagine then that if you have sheep that are able to have a reproductive life cycle of years, if you were able to find just from trial and error, oh, I got a white producer here, and I got another white producer here, guess what? These two, you guys, you're a pair. You are basically going to be mating for the rest of your life. Why? Because you know you got two sheep that have the capacity to produce white. And so you kind of maximize that. And so that's kind of, you can see it's the same basic thing. So what he was doing back thousands of years ago was basically the same thing that Mendel was doing. The difference is Mendel kind of laid this sort of veneer of scientific reasoning and questioning over on top of it, as opposed to just getting wool.
I can't believe even a quadratic formula existed back then.
It did.
How do we know that stuff so far, but yet they were still dumb in so many ways?
We are powerfully dumb in a lot of ways, actually.
Yeah.
But yeah, actually, that's the reason I love that is because when you take a look at that, you can see like that's like algebra, which we never thought was ever useful, but there it is right there. And Mendel's got it, you know, and none of that, but a lot of people don't realize the quadratic formula was developed by the Greeks to solve problems that they had at that time. We just lost contact with what those problems were, and now we have the formula left over. It's the reason why, honestly, math seems like a big Sudoku game, because we still have all the calculations and the formulas left over, but we've long since lost the reason why we developed them in the first place, right? And that's the reason why most people hate math, is because it's like a parlor game. It's like a parlor trick, but that's because math as a discipline has been horrible at actually preserving their own discipline. So that's because, and that's the reason, because when we look at math today, as we develop it today, we don't just develop math for kicks and grins. That we're developing math, like calculus is a good example, right? You develop that for a reason, because you're trying to solve some sort of a problem, and you have to create this math in order to solve it. That's the way we actually use math. Which is the reason why a lot of people like geometry a lot of times, or trigonometry, because it's like you're actually solving problems. You're looking at trajectory and angles, and you're trying to figure some things out. And that's why a lot of people are attracted to that sort of thing, because it's like, oh, hey, math at work. It's actually solving problems for me. Well, it actually all solves problems. Even factoring solves problems. It's just we've forgotten what those problems were, because they're way back in pre-Roman times when they got developed. So some of the basic conclusions. Ultimately, there's no intermediate traits. Sorry, Snapdragon fans, not there. Clear sense of dominance in this one, and of course, you got your 3-to-1 ratio, right? So that was a screaming factor for Mendel. This also leads us to kind of a 5-point model of that Mendel saw. We'll kind of just sketch this out a little bit, and we'll kind of fine tune a little bit as we kind of move forward. The first one is that these traits are basically discrete factors, right? So these are these particles of inheritance, these instructions, if you will, of inheritance, which we now know are genes. And that you'll only get one from each parent, one from mom, one from dad, that you've got alleles, which are alternative versions of the same gene. You've got homozygosity and heterozygosity, which are different types of genotypes, right? So these are all basically kind of come together to form the basis of what Mendel sort of described as sort of the way things are inherited, right? That's basically what he's trying to figure out, is how are these things actually inherited? And this is kind of like your 5-point model. But the problem is, what if it doesn't work this way? Well, we'll talk about that here in just a few slides. But before we get there, there's really 2 major principles or laws, if you will, that Mendel is credited for. The first one is the law of segregation. Now, basically, the law of segregation simply states that the two alleles, right, of your particular gene are basically segregated during gamete formation, and you get one from each parent. Now, a couple of things. First of all, this is a bit of a conflated slide. This is conflating what we now know with what Mendel did, and it's all basically getting mixed up together. But in order to divorce it and kind of keep it straight, Mendel, first of all, didn't know anything about gamete formation. He didn't know anything about gene segregation, like mitosis, meiosis, didn't know any of that. He didn't know anything about meiosis, didn't know anything about chromosomes, didn't know anything about any of that. Remember, all he's looking at is information flow. So what does he know then? Here's what he knows. I've got a paintbrush and I'm picking up pollen off of a purple plant. That's my male donor. I know that whatever that is, that pollen must have the information in it. Why? Because the information seems to follow that little gold dust. So whatever that is, purpleness is inside that gold dust. When I add it to the stigma of the female, then that adds it to her information. So whatever her information is must be in that little stigma somewhere. So what does that mean? That means the information for purpleness is segregating along with this gold powder, the pollen, from one parent to this process. And all of that, but the information for whiteness is in this female structure of the flower. So there's segregation of the information with the actual physical structure. He didn't know anything about meiosis, didn't know anything about mitosis, that came later. Didn't even know about chromosomes. Now we know that genes are found on chromosomes. That actually didn't lock into place until probably 20 years deep into the 20th century. So we're still not sure exactly what the inheritance molecule was for a while, for a good couple of decades. So that certainly wasn't around when Mendel was around. Which is one of the reasons why to come up with the law of segregation was an enormous act of intelligence on Mendel's part. To make that connection as significantly as he did is a big deal. It seems like a yada moment to us because we've got the hindsight in our favor. But most of the discoveries that kind of lock this into play and make this a yada moment came after Mendel. Mendel made this discovery and had this idea in his head before anybody knew anything about chromosomes or meiosis or mitosis, which is crazy, right? That just kind of goes to show you how ahead of his time he was, which is probably the reason why he got forgotten. But anyway, so then after this, and there's a little bit more, but what happened was Mendel published his paper on his results, and we got one more piece of result to cover a little bit before we move on completely from Mendel. But he published his paper in about 1865 or so. And what happened was his paper was largely forgotten for a good 40 or so years until like the turn of the 20th century. And then folks rediscovered it. So there was a group of geneticists like William Punnett, for instance, William Bateson and others that rediscovered his papers, and they looked at his results. They were fascinated by his results and what he got. And then the first thing they started doing was they basically asked the question, what Mendel did, will it work in my system? I'm studying corn. So if I do the exact same experiment that Mendel did, will I get the exact same information? Right. Some people have 4 o'clock plants. Some people had other types of organisms. They all asked the same question. If I do what Mendel did, would I get the same results? In some cases, the answer was yes. In some cases, the answer was no. Now, to the people for whom the answer was no, there was usually 2 responses from them. The first response was, oh, great, then Mendel was wrong. I knew it. He has no clue what he's doing. It's bad. It's garbage. And then the other group said, you know what? Maybe what Mendel was seeing was kind of like a pea plant thing. Maybe there's a different way of doing it in my system. And then they start going to work trying to figure out what that difference is. The response of the people of the former, those people were largely forgotten by history. It's the folks in the latter group who got curious about their own system, who wanted to take a look at the differences, like, why am I not getting the same thing Mendel got? Let's see what's going on in my system. It's different from Mendel. Those are the people then we go on and we know their names, which is always I use as a kind of a cautionary tale in science, is whenever somebody does something that doesn't agree with your data, don't ever, ever disparage them like that. Because almost always you will be forgotten by history. Because generally speaking is when you respond to your colleagues that way, is you are responding from a place of arrogance and insecurity. It's the people who say, you know what, I didn't get what you got, but I'm going to try to figure out and understand what you got so I can dig into my system and try to figure out what the differences are. And then on the way there, I'm going to make some pretty important discoveries in my own system, and then my name will be known. If I just balk at you and say, you're wrong, see, I didn't get the same thing as you did, so you're wrong. Good luck with that, because nobody will know your name ever. Right. And that's true. To this day, every single person that we know whose name is in a genetics textbook, there's, you know, a lot more, there's a lot more names in the genetics textbook. There's like all, they're everywhere. But not a single one of them had the former response. All of them were like, huh, what's going on in mine then? So if this is happening in Mendel, then how can we use what he did and try to figure out how to crack open what I'm doing? So they use it much more constructively. So then Punnett was one of those positive ones where he basically tried to want to figure this out. And these were, by the way, this generation that rediscovered Mendel was the one that came up with all those terms as well. They came up with all the labels like gene and allele and homozygous, this and that and the other thing. So they're the ones who came up with the actual terms that we now know. So now what Punnett basically did was create what's called the Punnett square, which is an organizational tool to help you define from any given mating what your potential outcomes are. So for instance, if you were to take a cross between two heterozygotes, the first thing you have to do is basically lay out which gametes each of them can produce. So for instance, this individual can either produce a gamete that's carrying the large P or the little p. This one, the same thing, large P or little p. Now, if these two gametes mated together, you would get big P, homozygous dominant. If these two mated together, you would get big P, heterozygous. If these two mated together, you would get big P, heterozygous again. If these two mated together, you would get little p, homozygous, recessive, right? Now, if you're something like corn, right, where you have lots and lots of progeny all at once, then you will see all of these classes represented. If you're like humans, where we only have one at a time, then typically our offspring will be one of these four. Okay. And so like, that's the reason why in human genetics, we don't say like, oh, well, how many offspring are going to be homozygous recessive? Oh, 25%. It's like, no, what are your chances of your one offspring being homozygous recessive? We have a 1 in 4 chance, right? So you have a 25% chance of your child being homozygous recessive. Right, so it's kind of different. It's a little bit of a slightly different twist on it when you get to the humans because we only have one at a time. But the Punnett square has been remarkably useful, right, to basically kind of figure out how to actually organize this. And so here's kind of what it looks like with their Punnett square. So we can see here's our parentals. So you've got your 2 homozygous recessives, and then your 2 dominant homozygous dominants. So you got your 4 heterozygotes, so all purple. And we cross these guys together, you got your big P, little p, and you're going to create your homozygous dominant in one case, heterozygous in the next, homozygous recessive. Notice you get your 1:2:1 genotypic ratio with your 3:1 phenotypic ratio. Now, the reason why this is important is because when you take a look at this, what you actually have is something that's predictive. And this is the reason why you know that this is right. So when I was at UC Davis and in a fly lab, one of the things I would do is you kind of set up this big fly screen that you would do. It's kind of big mating screen that you would do because you're trying to get some particular genetic outcome. And so you'd write it all down on paper. So you say, well, if I take a fly of this genotype and cross it to a fly of this genotype, then these are the potential outcomes I'm going to get in these proportions. And since flies lay hundreds of eggs, you're probably going to see all of them, right, in their relative proportions. And you're like, well, this is the one I want, this relative, just really, really rare exception. So I just want this one right here. And then I'm going to set her apart because I want to mate her specifically to another genotype. And so you kind of mate this one and you kind of, again, you sort of write out the cross, and these are all your different options and your relative proportions. And you kind of take this one, and you kind of do this for as many generations you need to go in order until you finally get your final set of data. But that's what I did before I ever crossed a single organism. So you'd write out this big mating scheme. And guess what? When you actually mated the flies, they did exactly what you wrote out. That is actually an important point to make, because what you've got here with your allele setup is you've got your theoretical, but your theoretical is so accurate that it can actually predict your actual, right? Anytime that your theoretical is so correct that it predicts your actual, then that means you are spot on. Or a lot of times that's missed by a lot of people. But that's exactly what you have this. And that's the reason why your allele concept, your allele framework of thinking is actually accurate, is because before you even put a single seed in the ground, you already know what's going to happen for generations out, because that's how tight that allele theory is. You can predict what's going to happen before you even plant the first seed. And that's what we can do in genetics. Now, the next big thing that Mendel did was a dihybrid cross. So we knew this was coming, right? Because we already knew the dihybrid corn existed. So what are we doing here? So you're taking two separate traits now in a single cross. You're looking at two of them. In this case, we're using R and Y, which is round yellow and wrinkled, round wrinkled yellow something. Red, yes, thank you. I always want to go purple, but red. Right, and so you're going to be basically crossing these in a dihybrid situation. And then ultimately, you're going to create in your F1 your heterozygotes. Notice it's what's called a compound heterozygote. Your heterozygotes for both genes, R and Y. And then when you cross these guys together, you're going to get a common ratio of 9-3-3-1, right? Where you have round yellow, round green, wrinkled yellow, wrinkled green. So 9-3-3-1. Okay, wait. Let's make some sense out of this first of all. I was going way too fast. Let's slow it down and let's add some more slides. Genetics has a lot of added slides in it. Mostly because there's a lot of unpacking that has to happen in genetics. Okay. Let's take a look at the dihybrid cross. We're going to start off with our parental. We start off with the same place we start off with before. True breeding strains. In this case, what I'm going to do is I'm going to use A and B, mostly because I can differentiate between the upper and lower case, right? So big A is going to be the dominant phenotype for whatever A is, right? Little a is going to be the recessive phenotype. Big B is going to be the dominant phenotype for whatever B is. Little b is going to be the recessive phenotype. So I'm going to basically take big A, big B, big B. That's going to be my true breeding strain. But remember, these two genes basically are understood as having nothing to do with each other. We're just happen to be tracking them. It could be A and Z for all we know, right? It could be completely unrelated. But remember that for being true breeders, you could also be a true breeder if I did little a, big B, big B. That could also be a true breeder. Why? Because they're homozygous for each gene, right? Or it could be little b, big A. That's also a true breeder. It's a true breeder for the recessive trait in B, a true breeder for the dominant trait in A, right? For the sake of clarity and simplicity, I'm going to stick with the top one. And I'm going to mate that with a true breeder, it looks like that. So when these guys cross together, I'm going to create an F1 that is big A, little a, big B, little b. And then I'm going to cross that, self it really, to each other. And then what I'm going to create is the F2. Okay. Now, building collapsing right at our ears when we're actually trying to do work. So the first thing is figuring out what gametes are produced by each, but they're the same, so they're going to be the same pattern, right? So they're both what we call these compound heterozygous. That means they're heterozygous at each gene, A and B. Okay, so first of all, if we take a look at the gametes, remember a gamete has to have a representative from each gene. So that means you have to pull an A, and you have to pull a B, and you have to pull a C, and a D, and an E, and an F, and all the way to Z, to however many genes you have, okay? So when you take a look at the A gene, you have two potential possibilities, right? You can either send the big A forward, or you can send the little a. So let's go ahead and take the big A. What if I send the big A forward? Well, I can pair this up with either a big B, that would be one gamete, or I can pair this up with a little b. That would be another gamete. But what if instead of grabbing the big A, what if I grab the little a? I could do the same thing. I could grab a little a with a big B, or a little a with a little b. Those are my four gametes that I can produce. As it turns out, because they're the same, the other parent does the same thing, right? So basically, big A, big B, big A, little b, this one and this one. There's my Punnett square. Notice how much bigger it got. I'm going from 1 to 2 to 3. Guess how many squares you have? 64. So you can see that Punnett squares are helpful as long as you keep the number of genes low. If you're looking at trying to analyze all the genes in a genome at once, it gets way too big, right? So it's got limited use, but it's good for a gene or a couple of genes, or maybe even 3 at most, if you want to just give that to an undergrad and drive them crazy, right? But you're probably not looking at 10, 12 genes all at the same time, right? So let's go ahead and take a look at this. Let's fill out all these possibilities, shall we? Okay, brace yourselves. So if we take the first two and we pair this one with this one, then we're going to get big A, big A, big B, big B. If we pair the other two, it's going to be big A, big B, little b. That's going to be big A, little a, big B, big B, big A, little a, big B, little b. It's going to be big A, big B, little b. Big A, little b, little b. Big A, little a, big B, little b. Big A, little a, little b, little b. Big A, little a, big B, big B. Big A, little a, little b, big B, little b. Little a, big B, big B. Little a, big B, little b. Big A, little a, big B, little b. Big A, little a, little b, little b. Little a, big B, little b. Maybe we don't want to do that one. You guys did that already in lab, didn't you? Yeah, you filled that one up. Maybe took you a little bit longer than that, but you guys did that with the tall and red exercise that you guys did. Okay, now let's analyze the phenotype, shall we? Okay, so first of all, remember we got A, right? So which is a dominant phenotype, and B, which is a dominant phenotype, that is capital A and capital B. So if I take a look at this guy right here, what's his phenotype? So it's going to be homozygous dominant on both cases, right? So it's going to be dominant phenotype in A. dominant phenotype in B, right? So we're gonna have a double dominant, dominant and dominant in both. So there's one. How about this guy? Same, right? How about this guy? So there's another one. How about this guy? Okay, yep. Next row, this one. How about this one? So this one's different, right? So now you have dominant in the first, recessive in the second. This is gonna be dominant, recessive. So there's one of those. How about that guy? So that'd be a class 1, right? The first class, this guy right here. So that'd be second class, right? How about this guy down here? So that's first class, this guy down here. Here's that one, this guy right here. So different now, right? We got a new one, this is class 3, recessive in the first, dominant in the second. So it's this one now. How about this one? So that's this one here, right? This one here. So that's the top one, yep. And this one. So that's class 2. This guy right here. So that's gonna be class 3. This guy right here. That's a new one, right? So double recessive. What's my ratio? Exactly. That's my phenotypic ratio for a dihybrid cross. Why is this important? Because this will only happen if your two genes are randomly segregated. As independent events. So what that basically means is that what's happening in gene A has no bearing on what's happening in gene B. They're independent events. So that's the only way you can get 9331. And that's actually kind of important because basically what Mendel then just found was not just what you saw the glossary from the first monohybrid cross, but not from the dihybrid cross. Now what he's saying is that, listen, this stuff is actually randomly segregated. So this is the first time we've actually started injecting this idea of the randomness of this, right? We've been talking about this all along, right? In meiosis, we've been talking about the random number generation, right? The associated with recombination and independent assortment and things like that, right? So we've been talking a lot about that. So we've been talking about the importance of this sort of randomization in order to create variation. But this is the first time that actually Mendel mentions this, that the genes themselves are segregating randomly. Okay. And that's kind of a big deal. By the way, this also flies in the face of what Colruyt already put to bed in direct transmission, right? Because not only is it not direct transmission, but the transmission that's actually happening is randomized. It has to be, because otherwise you're not going to get that ratio. So that's the dihybrid cross. And this kind of shows you what this looks like. And this is the one thing I don't like about this textbook. It's a little early, but I understand what they're trying to do. So we'll just do this. Because Mendel had no concept whatsoever of chromosomes. None at all. But what this is basically taking a look at the end of the 20th century and superimposing this on top of Mendel. That's basically what this is doing. So we obviously know now that a gene is basically housed on a chromosome at a specific location called a locus. So a gene's locus or a loci is basically where it lives on a gene. There's actually a physical spatial zip code, if you will, where the gene actually exists. We can actually map it. We can go to it. We know exactly where it is. Okay. So it's got a place to live on the gene. And that's kind of what they're trying to show you here. So here you can see you've got your true breeder for R and Y, basically given by your R and your Y, right? And of course, your true breeder for recessive traits. So here's your recessive traits. And then you generate your gametes, and then you create your compound heterozygotes, basically, in this particular situation. So you can see you've got a copy of blue and pink and green and yellow. So that's your heterozygotes. Then these are the gametes that you produce for your F2. And this is your dihybrid cross. So here you can see all of your round, wrinkled, and red and green, or excuse me, yellow and green. But you can also see the individual chromosomes that are going to be in these gametes as well, right? So understanding that all Mendel really had was just this here, this the information that he had. Later on, I'd say probably well into the 40s or 50s, that's the 1940s and 50s, this is when this came into play. So this is mid-century. Probably more like 50s, actually, when we started getting a handle on this. But they want to just basically make sure they tether in your head that this actually is the role of chromosomes, right? The reason why we talk about chromosomes, why we talk about chromosomes being important in mitosis and meiosis, is because this is where the genes live. They live on your actual chromosomes. So we've been already talking about that as sort of a yeah, that moment anyway, right? I mean, from the beginning, we talked about that. Okay. So now you've done it, haven't you? You've opened up the randomness now. You've let that genie out of the bottle. So what does that mean? That means you're going to now talk about and deal with the statistics, right? Because whenever you talk about randomization or random events, you have to talk about statistics. And this is kind of like one of those kernels in Mendel's paper that you see beginning to set up that entire statistical field of statistical genetics that gets very deep and started on really went cuckoo on that one in the 20th century. And you can do a lot of predictive genetics actually with a lot of statistics. And actually that served us well, because from that statistical approach, which gets very dense and very difficult, it's probably one of the harder branches of genetics, I would argue, is kind of wading through all that statistics and trying to make sense of all everything. That actually became a very necessary tool for human genetics, because we can't just cross ourselves off and generate progeny and analyze us. We kind of get what we get because families already are married and have kids. And we have to use a lot of statistics in order to predict whether or not, say, for instance, a particular family has a likelihood of a particular disease, a genetic disease, right? And that's kind of the way it works in human genetics. Is if somebody comes in and says, what's the chances that I'm going to have this horrible disease that's running through my family? Well, I don't know. Let's go ahead and take a look and see what it looks like. Right? We got to do some study of your family. We got to build out a pedigree. We got to see, which does Uncle Fester have it? Does Crazy Aunt Harriet have it? Do your grandparents have it? We got to figure out who has it, who doesn't. We got to get real. We got to have some hard conversations. Were your parents siblings? It's fun, right? But you have to know that, right? Because that affects and screws up the genetic pattern. That's need to know information from a human genetics point. Almost no family would admit to that one, except for the ones that make CNN every now and then, right? But that's kind of, we have to use a lot of statistics a lot of times. Like a lot of times we'll run into every now and then a particular individual who will have they may not have the disease, but they'll be a carrier for the disease. And they'll want to know, it's like, well, I don't know if my mate has this particular disorder, or if they have, or they're a carrier for this disorder, because they won't come in and get tested. So what's the likelihood of that? Well, let's take a look at this. Like, what ethnicity are they? We have some frequency data. We have some population genetics data from different populations. So we know, for instance, that sickle cell anemia is higher in frequency in African-American populations than in European populations. We know that there are other genetic disorders that are much more frequent in European populations than in African-American populations. So we have numbers actually from population studies of what the overall population frequency is. So if we know that your mate came from a particular ethnic background, we could use those population frequency statistics to kind of get an estimate of what your likelihood chance is that your partner is going to be a carrier of this particular allele. And that kind of gives you a little bit of information as to what the risk factor would be, So you can do that. But basically there's two rules. There's lots of rules, by the way, in statistics. We'll really focus on two. The first one is the rule of addition. The rule of addition, you're basically taking two independent events, two mutually exclusive events, and you're asking for a particular outcome to happen in either one or the other. So what you're looking for is you have two different events, A and B, and you're asking for an outcome to happen either in A or in B. And the important thing to understand is that or is your word trigger for addition. Right? Think back to your math, right? When you're learning about how to decode word problems, right? Certain words were basically a code for a mathematical operation, or is the word code in word problems for addition, right? So here's how this works. So imagine you have basically two coin flips, A and B, right? Those are your two events. And all you want is a heads. You want a heads in either A or in B. You don't care which one. You just want A heads, right? I just want an A or B. So the fact that it doesn't matter which one you get basically means that it is the addition of those two individual probabilities. So what is the probability of getting a heads in A? 50%, right? What's the probability of getting heads in B? Same thing, right? So then the probability of getting it heads in either A or B, either one, is the addition of those two probabilities. That's the rule of addition, right? So if you want to take a look at it in terms of genetics, we're going to take a look at our F1 cross. In this case, what we're asking ourselves is what is the probability of obtaining heterozygosity, of being basically a heterozygote? Well, there's two different possibilities here, right? Because ultimately, in one case, like in this case, you could get a big P from dad. What's the probability of getting a big P from dad? Half, right? Because his alternative would be little p, just like the quarter. It's either heads or tails. In this case, the big P or little p. So the probability of getting big P is 1/2. What's the probability of getting the little p from mom? Also 1/2, right? So in this particular case, the probability of getting big P from dad with a little p from mom is going to be 1/4. So what's the probability of the other one? What's the probability of getting a little p from dad instead of the big P? And then a big P from mom instead of a little p? So it's going to be the same thing, right? So it's going to be 1/4. So notice what you're asking for. Here's your either or statement. What's the probability of getting a little p from mom or dad? That's your or statement, right? Because if you get it from mom, then you get the big P from dad. If you get it from dad, you get the big P from mom. Either one of those is okay, right? It's either one or the other. So what do? You basically take your individual probabilities, which are 1/4, and you add them together, which gives you a probability of 1/2. Now you can also see this in the Punnett square, right? Because when you flesh this out, you're going to have your homozygous dominant, yes, your first heterozygote, your second heterozygote, and then your homozygous recessive. Notice what you have. 2 heterozygotes out of how many options? Out of 4. You reduce 2 out of 4 to what? 1/2. So you can see the Punnett square can actually show you visually what the actual probability is telling you mathematically. Again, nice correlation. You can predict one with the other and vice versa. That means it's a strong relationship. So the second major law that Mendel came up with from all of his dihybrid work and his probability work is the independent assortment concept. Now, basically what he saw here was that essentially that these guys are assorting independently or randomly. That's basically what we saw. Not only are they segregating, right, that your instructions, your particulate instructions are segregating into the individual gametes, which you didn't know what gametes were. This is like the pollen grain and the seed, probably what he called it, right? So that's the segregation piece of it that's going with that. But the other thing is that there's a lot of randomness to this, that there's random segregation as well. Segregation is happening, but it's also randomized. Now, the last rule of probability is rule of multiplication. So in this particular case, you're taking two independent events. In this case, you're asking a slightly different question. You have your two events, A and B. But now what happens is you want a particular outcome to happen in A, and you want a particular outcome happening in B. So in this case, you want A and B. Now, and remember is the word trigger for multiplication. What is the product of 2 and 3? Right. There's a couple trigger words in that one, isn't there? Product is multiplication, right? But 2 and 3 means 2 times 3, right? So just like, what is the probability of A happening and B happening? So if you were to take a look at the quarter method, basically you would say, what is the probability of flipping heads in A and flipping heads in B? In this case, it would be the product of the two individual probabilities. And so here you have, basically, if you're looking at homozygous, like generating the homozygous recessive, we've already established, first of all, that the probability of getting a little p from dad is what? 1/2, right? And the probability of getting a little p from mom, also 1/2. So what is the probability of getting one from mom and one from dad? It's 1/2 times 1/2, which is 1/4. Oh, by the way, let's draw out the Punnett square, shall we? Let's see if we can actually show this. So if we cross these, there's your homozygous dominant, yes. Heterozygous, heterozygous, and homozygous recessive. How many homozygous recessive do you have? Out of how many options? Out of a total of 4, and your probability is 1/4. So again, a nice connection between the Punnett square being able to predict what your probability is for your individual probability. So you could do like a couple of different ways. Okay, so another common thing that we use in genetics, and I can guarantee that Mendel probably had used this as well, was called the idea of the test cross. So what is a test cross? Well, here's a test cross. Basically, it's a situation where you have a dominant phenotype A, but that's all you know, right? So the idea is, what is this? I don't know. It could be one of two possibilities, can't it? It can either be this, or it can be that. So how do you determine the difference? What you do is you do a test cross. So essentially what you do is you cross it to its homozygous recessive. So essentially what you're going to do is you're going to set up yet more Punnett squares. And so your homozygous recessive is only going to have the homozygosity there, right? So it's going to be like that. Now this guy is either going to be this one, or he's going to be like this. Okay? So let's see what you get from either one of these. If it is a big A allele, then what's going to happen is you're going to get something that looks like this. Notice they're going to be 100% A dominant, right? Dominant phenotype, whatever that is. However, if that allele is the recessive, then you're going to get something that looks like this. 1-to-1 dominant versus recessive phenotype. So if you see any of that recessive phenotype popping out, then you've got yourself a heterozygote. So a lot of times students will ask, you know, the question, well, why use a recessive, right? Why homozygous recessive? Why can't we use a homozygous dominant? Why can't we use heterozygous maybe? And the reason is because, first of all, you can't use the dominants because you run into the same problem. You don't know if they're dominant homozygous or dominant because they're heterozygous, right? So you just compound the problem. The only way that you can know exactly what the alleles are is if it's homozygous. You know that because you only have those two homozygous alleles, that there is nothing else in there. You have no other thing in there. So the only differences that you see are attributable not to your test cross, but only to this guy. Why? Because you know what that's going to look like. You know what that's going to look like. You know what that's going to look like. The only differences that you see coming out are associated with that unknown allele. All dominant. Yep. So the other one you should see like a 1-to-1 ratio. And so this is kind of what it looks like. So here you can see, this is going to be, if it's homozygous dominant, it's just going to create a bunch of heterozygotes, and that's all you're going to see. If it's a heterozygous individual, then you're going to see this 1-to-1 relationship. And you can tell very quickly whether or not you got a heterozygote versus homozygous dominant. We actually use that quite a bit in genetics lab because a lot of times as we're working with different organisms, we'll see the dominant phenotype come out, but we're like, is that homozygous dominant or is that heterozygous? And so all we have to do is just do a quick test cross and we can figure that out. And then we can separate them out. You can also do this on the dihybrid level. So for instance, here you can clearly see these guys will breed true for both A and B. All right, this one will only breed true for B, not for A, because it's heterozygote. This one will breed true for A, but not for B because it's heterozygote. And this one won't breed true for either one, right? Because it's heterozygous in both. So you can do this on the dihybrid, on the trihybrid, if you wanted to do that kind of punishment to yourself, which I would suggest not. By the way, we don't even do trihybrids in genetics. It's just too labor intensive. By the time you get up to about your 20th, 30th square, you're about ready to jump off a bridge, and you're not even a third of the way there to fill in your 64 grid. And it's just like, and then once you get that phenotypic ratio, it's like huge and long. You're like, what in the world do I do with this? Well, nothing. That's the reason why we don't do it, typically speaking, right? There's other ways, more efficient ways that we've come across than just Punnett squares to analyze some of this across multiple genes. But okay, now let's kind of fast forward a little bit and kind of get to where we are, get ourselves current a little bit, right? So now we know, obviously, the DNA is the bomb, right? So DNA is our information. Oh, by the way, that was not always clear. So we spent a good chunk of the 50 years of the 20th century trying to basically prove what the genetic organism, what the genetic molecule was. Long time we thought it was protein. So that did not come overnight. This was after a good few decades of work that we got there. What we now know, obviously, is the DNA is coded sequence, right? And that it encodes the amino acid sequence that will allow us to be able to select the correct amino acid sequence to build our proteins with, a process that we will actually try to look at in a couple of chapters, in chapter 15. Now, we also then have the ability to alter that, right? So what happens if we alter that DNA sequence? Then that's what happens if you alter your DNA sequence, and you're going to also alter your phenotype, right? Now, you alter your phenotype by changing the sequence of amino acids, which then can lead to dead protein. And that could be a problem. Now, this is ultimately will then be like, if you do a mutation, this will be acted on by natural selection, which will always act on phenotypes typically. If you have a favorable phenotype, then you do well. If you don't, then you don't do so well. A mutation is one of the big mechanisms of evolution. Problem is, it's got some serious issues. Because here's the problem, right? I mean, the way kind of the Darwinian model goes, and I will admit, quite frankly, I'm not a Darwinian at all. I'm more of a punctuated equilibriist. I'll explain what that means actually in just a second. You're like, what? Lay that one on your friend, right? So I'm a punctuated equilibriist. No, but here's the reason, right? Because the idea is that basically all of the genetic modifications to our phenotype had to come had to come to us basically by way of mutation. And if you think about it, we're talking about a truckload of mutations that would have to be required in order to create a particular phenotype. And this gets even more compounded actually when you take the genetics class, because you realize just how difficult it is to basically put all this together. There's so many moving parts to it, and it all has to come together just right in order to create this phenotype, which looks to us to be fairly straightforward. I mean, when we're talking about dark hair, light hair, it seems very kind of Pollyanna, but it's not at all, right? Part of the problem is that mutation as a major mechanism of evolution is problematic because most mutation rates that are published are relatively slow. And at best, most of them actually are, it's even worse. It's actually even slower than we think, because it's not just mutation accumulation to drive evolution, but it's beneficial mutation. By far, the majority of these are deleterious. That means they kill you. And how do we know that? Because I've got a big old thick book, which is now actually from 88, actually. That's when it was last published, in a book. It's now online, of all the human genetic disorders that we know about. And that was like as of 88. It's now online. It's on Mendelian, it's online Mendelian inheritance in man. It's OMIM, basically. But it's a database now of all of the known genetic disorders in humans. It's a mind-numbing pile of catastrophe. And when you look at it up close to this, you're like, oh, wait a minute. So you're trying to tell me that mutations that created this mind-numbing pile of catastrophe, genetic catastrophe and human carnage, that's what's driving what happens here. That is a little far-fetched, because this was a problem, right? Because the mutation rates are so slow, and the beneficial rates are even slower than that. That you're talking about requiring not 4 billion years worth of evolution. You're probably talking about enough time like pre-Earth. You're talking about like 14 billion years of evolution would be required in order to accumulate this number and this collection of beneficial mutations over time. So it's got a lot of problems with it, right? So is it powerful? Yes. But does it have some really, really strong caveats? Yes. And I think we're foolish if we don't go in eyes wide open with those strong caveats in mind, right?
How long, let's say, would it take for the beneficial mutation to actually develop? Like, let's say we all develop a second part. Let's say that's beneficial, one stops, we continue. And how long would that mutates today, one individual? And how long would it take for us to be like the normal human?
To develop a new heart? So to basically develop a new heart, like if you were to just go with like one mutation at a time, you're probably talking about, I would say, hundreds to thousands of new mutations that you would require. Each mutation, just mutation rates in general can range. They're not all the same. There's different published rates, but usually they're in the 500,000-ish range to perhaps even 1 million sometimes in some cases. One mutation that sticks, that goes forward. That's not necessarily differentiating between beneficial and non-beneficial. So the mutation rates are catastrophically slow. And we know that, which is the reason why mutation rates are problematic. right? When you combine that with the fact that we actually struggle to actually come up with beneficial mutations, then the next obvious question is, well, then how do you expect enough beneficial mutations to accumulate to create this? I mean, think about this for a second. There's like a common sense there. And so this is basically actually what led to, and kind of the reason why the Darwinian model is insufficient, and the reason why it kind of fell apart actually in the 60s and 70s. And I say this as somewhat heretical because UC Davis is a Darwinian school. So you can imagine how I kind of to keep a low profile there, because they were Darwinian, hardcore Darwinian folks. They were wrong, but they were Darwinian folks. Yeah, I have no problem telling people they're wrong. I'm very opinionated that way. Right, but the idea was, this is the reason, part of the reason why Stephen Jay Gould and Stephen Elledge came up with this concept of punctuated equilibrium. And it was in part to answer the deficiency of the Darwinian model. Because remember when I said that genetics is quintessentially at the core of evolution. Evolution is a genetic concept, and that it kind of started off on a very strongly dysfunctional foot, because it did not start off genetically. It started off with puttering around with a bunch of rocks, right? Well, the problem is that fossils and morphology actually don't cut it. There is serious deficiencies in fossil record that we just couldn't get over. I mean, we've all heard of the missing link, right? It's not just a criticism, it's actually real. There are no links in there. Even the so-called missing links, like Archaeopteryx, have later actually been seen as fully fledged species that aren't really actually enter anything. They're actually their own thing. But the funny thing is, oh, guess what? We went to the presses with the missing link, but we didn't actually, we kind of whispered the retraction. It's like, oh yeah, by the way, we screwed that one up. Yeah. Right? But those deficiencies are titanic and ubiquitous throughout the fossil record, which is one of the reasons why I'm not interested in it at all. But it's also part of the reason why Stephen Jay Gould and Stephen Elledge came up with punctuated equilibrium. So their idea was that it's not just a Darwinian model, which basically Darwinian gradualism is resting on geology, which is kind of utilitarianism, which is essentially that all the basic slow creeping processes like sedimentation and erosion were basically in place from the beginning of time to now, and that this is basically kind of like a clock, that these processes go forward in perpetuity in the future without disturbance and without alteration. So that the rates of deposition and erosion that we see today were the same rates that we saw back in the Triassic, right? So we can basically say then, based on the rates, how long it's been since this mountain was formed. Hundreds, perhaps, multiple hundreds of millions of years ago. And so that's basically what Darwinianism is based on. There's one tragic flaw to that, and this is the reason why I reject uniformitarianism, is because originally it was set up to reject catastrophe. They basically said it's all uniform and it's all constant rates, and we do not have room in our model for catastrophe. Well, which is idiotic, right? Because we know that catastrophes happen, and we also know that catastrophes have the capacity to do rapid remodeling of the environment in a very short period of time. Mount St. Helens is a perfect example, right? I mean, you basically lost half of a mountain in a matter of seconds, right? You got entire canyons carved out of there by the big massive mud flows in a matter of weeks, right? So that wasn't just slow deposition over time with erosion, that was rapid catastrophic flows. The same thing happens with things like Chicxulub, right? Where you have this, that's the one that killed the dinosaurs 65 million years ago. When that one first came out, that was super controversial. People were like, no, nothing like that happened. The dinosaurs died slowly over time, right? Over hundreds of millions of years ago, that's how they died out. So we didn't really wrap our heads around this idea of a catastrophic event having an extinction level impact on the Earth until very recently. And that's what Gould and Elledge were saying. What they were saying is that, okay, most of the time you have this Darwinian kind of a stasis, this equilibrium. That's where their punctuated equilibrium comes from, where all the rates are the same and uniformitarianism seems to be at play and everything's very predictable. But then there's these punctuations of catastrophe in between. And these catastrophes are typically met with elevations of mutation rate, where you have these periods of rapid speciation, which happens so quickly. And typically the idea under that is that all these mechanisms, including mutation, increase in speed rapidly. So that you get this rapid speciation over relatively short periods of time. But because it's so fast, you don't ever deposit missing links, because you never stop at the missing link. You're always moving forward because it's change that is so rapid that if you don't look at it, you're going to miss it. And so all you see is the finished product in the fossil record. And that's all you see. And so that's what Gould and Elledge basically said. And it was a response to the deficiencies of traditional Darwinianism. Now, as it turns out, what we actually are starting to come around to is an amalgam of that. Some Darwinian features, some sort of long-term uniformitarian sorts of features with punctuation in it. So some systems will be better explained with this sort of Darwinian gradualism. Other systems are much better explained with punctuated equilibrium, right? So it's kind of it's becoming an amalgam of the two. And now the most recent addition to that is what I hope at some point is a replacement of dusty, dry rocks with actual DNA data. Is that a question going up, Blake? Oh, you mean like the meteors? Well, we know there's been a lot in the past because Chicxulub is only one of many, many impact craters. Several of them potentially instinct level. Chicxulub is only actually like the fourth largest, actually. There's been larger in the past.
So it was one that's not part of population. How long was
Oh, that's a good question. They could probably tell us fairly quickly because they're pretty good at being able to calculate the trajectory of some of these things.
once they figured out what they say, like it's 10 years, or it's gonna be like another 100,000 years? Or could it come up like?
It depends. There's some that they know very well and that they can calculate very well as they come in. But then there's others that seem to surprise them. Like, whoa, we didn't know this one was a thing. So they do get surprised. So, but I would say, even if they're able to notice it in 10 years, it doesn't really matter because we're screwed. We have nowhere else to go. And I can guarantee you right now, we're not going to be ready to go to Mars in 10 years. So it's either you die on Earth or you die on Mars. That's going to be your two options. Right. So that's really lovely, right? But that's basically what you've got, right? So you every now and then we can see this. You have like an extinction level event, like Chicxulub, for instance, which wipes out the entire dinosaurs. It's an extinction level event. But then you kind of have populations that go through a bottleneck. And when you go through a bottleneck, a lot of your mutation rates change. So a lot of the evolution, like the species, like speciation, changes a lot. So like, for instance, and that's not just with new mutation, new DNA, it's with existing variation. So a lot of times existing variation can move quickly when you have small populations that get isolated geographically. And so you can start to really tease this out. And you can see this a lot in like Darwin's finches, right? So where you kind of see these different species of finch sort of basically adapting and becoming new species, but it moves very quickly because they're responding to their environment. That happens much faster in small populations than in large populations. So that amount of variation, that amount of adaptation happens very quickly at first. So if we go through an extinction level event, which presumably there's evidence genetically that we might have gone through a couple actually, and we go to that bottleneck, then what comes out the other side is a population that with the existing variation they have, now they probably lost a lot of genetic information through that disaster, but what they have now, is going to adapt very quickly. So they're going to look almost like evolution, which is what it is. It's a speciation, right? But you didn't create any new variation. It was just endogenous variation where that well is deep. And so when you get those random number generators going quickly, then you have a lot of adaptive capacity. And that really gets teased out when you have a small population trying to adapt to lots of different ecological niches. We see that a lot in population dynamics. And that is an idea that I think is undersold in evolution. Is we talk a little too much about mutation, which is weak, but not enough about, okay, but once you actually have this variation in there, but I mean, you could probably start about the family level. There's already a lot of genetic variation in the family level already. You can see already just a lot of non-new DNA, just adaptive radiation. It's just like all you're doing is just shuffling and creating new variants, genetic variants, and each of them are taking to their environments. And that's what creates the downstream genus and species and subspecies. And that's kind of what's creating that bottom half of the tree. So there's not enough about that, which I think is important.
When it comes to like people in power, like really high up,
Mhm.
is it genes that makes them be so okay with the thin air, or is it just the body that adapts?
So that probably is, there is probably an adaptation there, a combination of both, right? The problem is if you're in Nepal, you probably only live there all your life. So everything about your physiological existence has been that. So you've never not known that challenge, right? I think, especially like base camp, I think is like 17,000 feet, right? That's kind of where you live, like normal. That's like your normal living, which is crazy, right? 17,000 feet. That's like even above the continental divide, right? By like a good 5,000 feet, right? So we have another mile-high city above the continental divide when you're talking, and that's just base camp for Everest, right? But there's like a whole little village down there. They are quite ancient. They go back thousands of years, right? So you probably do have some population genetics fixation of alleles in there that are beneficial. Why? Because if you're not able to cut it there, guess what? You're dead. If you are, you're going to live and you're going to reproduce, and your genes, which are robust for that environment, are going to compound, and it's going to increase in that population.
I can barely run out here. So.
Yeah. Yeah, just imagine what it's like there. Right. So yeah, so that you see that actually a lot. And this is the reason why population genetics is so fascinating, because you can see it in human populations. You can see it in medical epidemiology, right? So you can see the way like different viruses and diseases are sort of fluctuating around the world and sort of where they're coming from and how they respond to different groups of people on different continents. I mean, obviously the Italians didn't do so great with COVID. So it was really bad to be an Italian for COVID, but some of the other countries didn't get it so hard, even though they got the full force of COVID. It wasn't nearly quite like what the Italians were going through. I mean, they got ripped. I mean, big time. So there was some sort of a kind of a population genetics thing that just did not favor their genetic kind of alignment, the fixation of alleles that they have in the Italian population with the way COVID reacted. It's a perfect example of genetics, the importance of your genetics filter with respect to human healthcare, right? So it really does kind of define our existence on just about every level. Okay, now let's take a look at some of the extensions to Mendel. So what is an extension? Well, it's basically this is about when. So up to this point, this is what Mendel did. So now what we have is a go right about there. So basically, this is the dihybrid cross is about where Mendel stopped, and then that's what he published, the monohybrid and the dihybrid work, and then that's what got rediscovered 40 years later by all these different geneticists, and they were all working on different things and different systems. Like I said, some were working on corn, some were working on 4 o'clock plants, and some were working on humans. So all sorts of different things, and they all had the same question. Like I said, right? Does this work in my system or not? And then when those who we remember, right, the ones who said, oh, wait a minute, I didn't get the same thing Mendel got. So what does that mean about mine, right? So what's mine doing? Those are the ones who went on to populate the other half of this book. Because what they noticed was that Mendel saw a particular set of rules by which genes seem to be operating. And in some cases, those rules apply, but in some cases they don't. There's almost like a different set of rules. So I've got one of those. So the question is, of course, what are my rules? Well, what this is, the extensions to Mendel, is those rules. Each of those rules we refer to as patterns of inheritance. The rules by which genes will segregate. Now, the first one we've already covered, the Mendelian pattern. So what does Mendelian inheritance tell us? Well, there's 3, 4 maybe major criteria for Mendelian inheritance. The first one is Mendel saw one gene, one trait, right? Flower color was the function of the flower color gene. That was it. None of that, but what Mendel also saw was 2 alleles per gene. You either had purple or white. You either had yellow or green, smooth or wrinkled, right? There was only 2 alleles per gene itself. The other one he also saw was complete dominance. When the 2 were together, did you see the recessive? Never. Total blackout, right? Complete dominance. Oh, and the third one, or the fourth one, which we don't really have a pattern inheritance for this one, particularly, well, we do actually. I just want to lay it in there, but is random segregation. The reason why I like random segregation is because it combines both of his ideas. Segregation with randomness. Okay, that kind of encapsulates his laws. So if your system or your gene follows these criteria, then it is Mendelian. What should you expect to see? Easy. 3-to-1 phenotypic ratio, a 1 to 2 to 1 genotypic ratio, and if you're dihybrid, a 9-3-3-1 phenotypic ratio. Okay. Those are your diagnostic ratios. What's the importance of the 9-3-3-1? Well, when you get into different patterns of inheritance, and this is kind of something we kind of get into a little bit deeper in genetics, is all the different patterns of inheritance actually are based on this, the dihybrid cross. Because let's say, for instance, that you have 2 genes that are interacting with each other. That means they're not independent, right? That's already a violation of Mendel. Right? Mendel said these things are randomly segregating, but they're independent events. But if these 2 are interacting with each other, then what's going to happen is they're going to be segregating, and what happens in A is going to mess with what happens in B. When you have that kind of non-randomness associated with that, what it does, it creates different ratios. But those ratios are based on 9-3-3-1. Here's a good example. A good example is epistasis. We have a version of epistasis, which is 2 genes that interact with each other, so they're not independent of each other. But the ratio for epistasis is 9-7. 9. And then 3 plus 3 plus 1 is 7. So all these gene interactions are actually founded upon this 9-3-3-1 random situation. So I have sometimes I'll see 12-3-1, or I'll see 15-1. So you'll see lots of different 9-3-4. Sometimes you'll see. So you'll see different variations of this 9-3-3-1. Each one of those, just like 9-3-3-1 is diagnostic of the dihybrid cross, 3-1 is monohybrid cross. A 9-7 is diagnostic ratio for epistasis. So if I'm a geneticist and I have 2 genes and I'm studying them and I want to know what pattern of inheritance do these guys follow, what rules do these guys follow? If I cross them together and I get a 9-7 ratio, guess what? They're epistatic, right? If I get a 15-1, then that's another pattern. If I get a 12-3-1, then that's another pattern. So depending on what the pattern is, when I cross these 2 together, it'll tell me what set of rules they're following. And I'll have some understanding about how this pattern works in this particular population. So this is our first set, Mendelian. But now what I want to do is I want to start violating violations. I want to start violating each of these criteria, right? Deviations from Mendel. This is the reason why we have this. The first one that we're going to take a look at is polygenic inheritance. That's the first violation. So polygenic inheritance, basically what you've got is a situation where you have one trait caused by many genes. Polygenic, many genes. So when you have this, basically, it's oftentimes referred to as either a continuous trait or a quantitative trait. Sometimes it's also referred to as multifactorial, is an extension. We'll talk a little bit about that. So the reason why we say continuous is because when you have lots of genes involved, you're going to have lots of data points, right? So like an example would be human height, right? This is not categorical. You don't have people that are 4 foot, and then people that are 5 foot, and then people that are 6 foot, and then people that are 7 foot, right? You have everything in between, don't you? You have 4'10", 4'10" and a quarter, 4'10" and a half, 4'10" and 3/4, right? If you really hang on to those fractions of an inch. I did when I was a teenager. I gave up because once I realized I was losing that 1/4 of an inch, I just gave up and just said, yeah, I'll just stick with 5'10". But now I'm probably not even that. Now I'm probably like 5'2" or something like that. But anyway, I'm on the shrinking side of life now, right? But the idea is you have every data point in between, right? So that's basically what we call continuous. So when you have lots of data points in between, you don't just have a category, you have every single potential individual point. When you do that, it creates a curve that looks like this, with the mean in the middle and two extremes on the side. What do we call this? This is your bell curve. This in statistics also has a couple of names. A continuous distribution, which is why we call this a continuous trait. And it's also called a normal distribution. But this is the reason why this is called a continuous trait, because you've got, every time you add a new gene in there, you've got another data point to add in there. So you basically add more data points in between. This creates this kind of smoothing out, or this bell curving of your data pile. Okay. Now, one thing I want to do before I let go of polygenic is when you take a look at the polygenic, multifactorial traits typically are quantitative traits. That is to say, polygenic, but also they tend to have an environmental aspect to it as well. A good example of that, behavior. Behavior is equal parts genetics and environment, right? So that would be multifactorial in nature. And this kind of just shows you what we're looking at, right? Here's your height. Line up according to height. What are you going to see? You're going to see a histogram. You're going to see a lot of people somewhere between. You're going to see a few rare tall people, few rare short people, but most people are going to be somewhere in between, with every possible permutation in between the two extremes. That is a continuous trait. The more genes you have, the more that you have. Skin color is another example of continuous trait, because we have like 8 genes in skin color. They all have the same thing, dark allele, light allele. So when you inherit them, you just basically inherit either an accumulation of dark alleles, which means you have darker skin tone, or an accumulation of light alleles, which means you have a lighter skin tone. So generally speaking, when you have all 8 of these, it's very rare to accumulate all dark alleles, and it's very rare to accumulate all light alleles, which is the reason why very, very light people are rare, very, very dark people are rare. Most people have a mixture of dark and light somewhere in between. Most of the world is in that sort of middle-ish olive colored zone. And that's a bell curve. If you take a look at skin tone, it's a bell curve. Very few very dark people, very few very light people. Most people are somewhere in between. right? Kind of olivish, either olive dark or olive light. Okay, that's where most people are. So we'll take a look at pleiotropy next time. Go ahead.
No, no.
Oh.